You know how our immune system is this incredible bodyguard, right?
Mm,
Always on patrol, ready to take down invaders. It's really uh, quite something when you think about it, this defense network running constantly.
absolutely. It's incredibly sophisticated.
But what if it gets confused? What if, it mistakes a part of us for an enemy? What happens when that bodyguard suddenly turns rogue?
Well, that's precisely where molecular mimicry comes in. It's a fascinating, but sometimes truly devastating biological phenomenon.
Okay.
It's where foreign substances, maybe proteins from viruses or bacteria, they look so similar, structurally speaking, to our own body's proteins that are our immune system. It just can't tell the difference.
Wow.
Yeah.
Yeah.
So today, we're diving deep into how this biological misidentification can lead to autoimmune conditions. Specifically, we're looking at those affecting our nervous system.
The nervous system.
Exactly. We're talking about demyelinating diseases, conditions where the protective insulation around our nerve fibers, the myelin sheath, gets damaged, and that severely disrupts nerve signals.
Right. The vital communication pathway.
Precisely.
So our mission today, then, is to really unpack this intricate dance of mistaken identity. We'll explore the what, how, and why of molecular mimicry, especially how it connects to myelin, which is just so crucial for nerve function. Think of this as your shortcut to understanding a really complex but incredibly important area of immunology. We're getting an MD immunologist perspective here, aimed at helping students really grasp it.
I'll try to break it down. It's a journey into how our biology can take these unexpected turns. It shows the incredible sophistication, but also, frankly, the vulnerabilities of our immune system.
Let's get into it.
The immune system's double-edged sword of primer for students.
Okay, let's unpack this. Before we dive headfirst into the mimicry part, maybe we should do a quick refresh.
Yeah.
Just on the main players, B cells and T cells.
Good idea. Foundational stuff. They're like the special ops units, as you said, each with really unique roles. Constantly surveying.
So what are those roles exactly?
Okay, let's start with B lymphocytes, or B cells. They're the main players in what immunologists call humoral immunity, their big job, producing antibodies.
Antibodies, the Y-shaped things you see in diagrams.
Exactly those. Think of them as highly specific guided missiles. Each antibody is tailored, like perfectly designed to recognize and latch onto a specific foreign invader and antigen.
And once it latches on.
Well, it can mark it for destruction by other immune cells, or it might block the invader from entering our cells, or even neutralize toxins directly. Super important for clearing out bacteria or viruses just floating around in our blood or tissues.
Got it. Extracellular threats.
Precisely. Then you have T lymphocytes, or T cells. And these guys, as some of our background material puts it, are often seen as the true ringleaders of the adaptive response.
Wing leaders. Okay, so they're in charge.
In many ways, yeah. They're much more diverse than B cells. You've got different subsets doing different jobs. First, there are T helper cells, also called CD4 plus T cells.
CD4 positive. That sounds familiar.
Right. The CD4 is just a marker, a co-receptor, that helps them see antigens presented in a specific way on something called MHC class II molecules. We'll get to that. But these helper T cells, they're the strategists, the coordinators.
So they don't do the killing themselves.
Generally not directly. They orchestrate. They send out chemical signals, cytokines that activate B cells to make better antibodies. They rev up the killer T cells. They call in other cells like macrophages. They basically shape the whole immune battle plan.
Okay, makes sense. Who does the killing then?
That would be the cytotoxic T cells, or CD8 plus T cells. They use a different co-receptor, CD8, to bind antigens presented on MHC class I molecules. These are the direct assassins.
Assassins, strong word.
Well, it fits. They specifically hunt down and eliminate our own cells that have become infected, say, with a virus, or cells that have turned cancerous, or even cells just damaged beyond repair. Absolutely critical for intracellular threats.
So B cells for outside, killer T cells for inside, roughly.
Roughly, yes. But there's another crucial T cell type. Regulatory T cells, or TREGs. These are the peacekeepers.
Peacekeepers, in the middle of a battle.
Exactly. Their job is vital. They maintain immune tolerance. They prevent the immune system from going overboard or, crucially, attacking our own body. They act like a brake.
So they stop things getting out of control.
Precisely. Once a threat is handled, TREGs help wind down the response. And very importantly, they suppress any self-reactive immune cells that might be lurking around. So you see, it's this incredibly complex, multi-layered system.
It really is. So these specialized cells are out there. B cells, T helpers, killer Ts, TREGs. But how do they know? How do they get their marching orders? How do they distinguish, a harmless pollen grain from a deadly virus or from, us.
Mhm.
That recognition step seems fundamental.
It absolutely is. And that brings us to a really key process, antigen presentation. Our body has specialized cells just for this job. They're called antigen-presenting cells, or APCs.
APCs.
And among the most important, the most efficient APCs are the dendritic cells. Think of them as the sentinels, the intelligence gatherers.
Where do you find them?
They're strategically placed all over, especially in tissues that face the outside world: skin, lungs, gut lining, but also in organs. When a dendritic cell bumps into a pathogen, maybe a virus,
Yeah.
it doesn't just ignore it. It acts like a sophisticated vacuum cleaner almost. It gobbles up the pathogen, breaks it down into smaller pieces. These are the antigens, and then it displays these fragments on its surface.
Displays them, like showing them off.
Exactly like showing them off. It uses special molecules for this display called major histocompatibility complex, or MHC molecules.
MHC, I've definitely heard of that. Class I and Class II.
You got it. Two main classes. MHC class I molecules are found on almost every cell in your body that has a nucleus. They usually present antigens that come from inside the cell.
Like if a virus gets in.
Perfect example. Or if a cell becomes cancerous and makes weird proteins, these MHC class I complexes basically wave a flag to the CD8 plus cytotoxic T cells, saying, hey, I'm compromised, eliminate me.
A kill signal.
Essentially, yes. Now, MHC class II molecules, they're mostly found on those professional APCs, dendritic cells, macrophages, even B cells. They present antigens that came from outside the cell.
So something the APC ate.
Exactly. Maybe a bacterium it engulfed. These MHC class II complexes show those fragments to the CD4 plus 2 helper cells, and the T helper cell then figures out, okay, this is an extracellular threat, we need antibodies, or maybe this needs macrophages activated. It orchestrates the right kind of response.
So the MHC system is how the T cells know what the threat is and where it came from inside or outside the cell.
You've nailed it. It's incredibly elegant and ensures the response is specific and appropriate.
And you mentioned dendritic cells are also balance keepers. What's that about? It sounds like more than just presenting antigens.
Oh, absolutely. Their role in regulation is pivotal. It's a masterclass. See, immature dendritic cells are amazing at just sampling their environment, constantly picking up bits and pieces, cellular debris, maybe harmless stuff, maybe pathogen fragments.
Without raising the alarm every time.
Right. They actually keep their MHC class II molecules kind of hidden inside, sequestered, until they get a real danger signal. Like they detect specific microbial patterns, PMPs, maybe through toll-like receptors.
Danger signal received. Then what?
Then they mature dramatically. They start expressing more MHC molecules. They put up other flags called co-stimulatory molecules, absolutely essential for fully activating T cells, and they travel to the lymph nodes where the T cells hang out.
So they go from quiet samplers to loud broadcasters.
Perfect analogy. They transform into highly effective T cell stimulators. They're the bridge between that immediate, nonspecific, innate immune response and the highly specific, memory-forming, adaptive immune response.
And that's the balance-keeping part, deciding when to sound the alarm.
Exactly. Their ability to accumulate antigens quietly, combined with this tightly controlled maturation process and their power to stimulate T cells, makes them critical deciders. They activate immunity decisively when needed, but just as importantly, they help preserve tolerance by not freaking out over every little thing, especially our own self-antigens. It's a very controlled process.
Okay, so the system is designed to hit foreign invaders hard. But the flip side, how does it stop itself from attacking us? With all these powerful cells and signals, it seems like autoimmunity would be happening all the time.
That is the million-dollar question, and it brings us squarely to immune tolerance. This is the fundamental ability to tell self from non-self, and crucially, to not attack self.
How does it work?
There are several layers. The first big one is central tolerance. This happens while B and T cells are developing in the bone marrow for B cells, the thymus for T cells. It's like boot camp.
Boot camp.
Yeah, they undergo rigorous selection. It's called clonal deletion. Any lymphocyte that reacts too strongly to self-proteins presented there gets eliminated. Apoptosis-programmed cell death. Bye-bye, self-reactive cell.
So the really dangerous ones get weeded out early.
Mostly, yes. But it's not a perfect system. Some self-reactive lymphocytes do manage to escape central dilution. They slip through the net and get out into the body.
Uh-oh, that sounds bad.
It could be, but there's a backup. Peripheral tolerance. This happens out in the tissues. There are several mechanisms here. One is clonal energy. If a self-reactive cell encounters its self-antigen but doesn't get the right co-stimulatory signals, because there's no danger signal around, it just gets shut down, becomes unresponsive.
So it sees the target but doesn't get the go signal.
Exactly. And then critically, we have those regulatory T cells, TREGs we talked about. They actively patrol and suppress any self-reactive cells they find trying to get activated. They are absolutely key peacekeepers in the periphery.
So even though some self-reactive cells exist, they're usually kept in check.
Usually. But the fact that they do exist, these residual self-reactive cells, well, that lays the groundwork for autoimmunity if something disrupts that regulation or if something activates them strongly enough.
Right. And speaking of regulation, let's circle back to those T cell subsets. We mentioned TREGs, but you also got up Th17 cells. How do these different types fine-tune the response, especially around this tolerance issue?
It's an incredibly complex symphony, really. Beyond the general helper function, these CD4 plus T cells differentiate into distinct subsets, each making a unique set of cytokine signals that drives a specific type of immune fight.
Okay.
So Th1 cells, they're big on cell-mediated immunity. They crank out interferon gamma, IFN-day. This stuff is great for activating macrophages to gobble up intracellular pathogens like viruses or certain bacteria. Really ramps up the cellular defense.
Got it. Th1 for cellular. What about Th2?
Th2 cells lean more towards humoral immunity, helping B cells make antibodies and fighting things like parasitic worms. They make different cytokines, like interleukin-4, IL-4, which pushes B cells towards making IgE antibodies, often linked to allergies, and interleukin-10, IL-10, which is actually anti-inflammatory, helping to dampen things down.
Okay, so different jobs, different signals. Now, Th17, you said they were linked to autoimmunity.
Yes, Th17 cells. These are the ones we often find are, let's say, overly enthusiastic in autoimmune diseases. They are highly pro-inflammatory.
How do they get started?
They mature under the influence of specific signals, particularly cytokines like interleukin-6, IL-6, and TGF-O. And once they're active, they pump out their own potent inflammatory signals, especially interleukin-17, IL-17, IL-22, and sometimes others like IL-1.
And IL-17 is bad news in autoimmunity.
It can be, yeah. IL-17 is really good at calling in neutrophils, those frontline inflammatory cells, to a site. In an infection, that's helpful. But in autoimmunity, it means neutrophils flood into healthy tissue and cause a lot of damage.
Ouch. So Th17s drive inflammation and tissue damage.
That's a key part of their profile in autoimmune settings. And this is where the contrast with TREGs is so stark. TREGs, remember, are actively suppressing immune responses, often using some of the same signals like TGF-β, but in a different context, plus IL-10 and direct contact.
So it's a tug of war between Th17 driving inflammation and TREGs trying to keep the peace.
Exactly. It's a critical balance. If that balance tips maybe too much Th17 activity or not enough TREG function, you're heading towards autoimmune trouble. A lot of research, including work mentioned in our sources, shows how disrupting this balance is central to many autoimmune diseases. It's why these pathways, IL-17, IL-6, TREGs, are such hot targets for new therapies.
Molecular mimicry, the art of deception.
So we have this incredible, complex, usually well-behaved immune system with all these checks and balances. Here's where it gets really interesting. How does it get this self and non-self idea so wrong sometimes, even with tolerance mechanisms?
This is the absolute core of molecular mimicry. It's where the immune system's amazing specificity, its greatest asset, actually becomes a liability.
So,
Simply put, molecular mimicry happens when there's a strong similarity, maybe in the amino acid sequence or the 3D structure, between a piece of a pathogen like a viral protein fragment and one of our own host proteins.
they look alike.
Very much alike. So much so that our immune cells, the T cells and B cells that were primed to fight the pathogen, can't reliably tell the difference between the foreign invader and our own tissue.
Oh, I see. So the attack continues but hits the wrong target.
Exactly. It's immune cross-reactivity. The immune response, which started appropriately against the pathogen, spills over and mistakenly attacks self-components that look similar. That's what drives the autoimmune disease in these cases. It's a case of mistaken identity at the molecular level.
A biological disguise, almost.
It really is. And it exposes this cruel irony. The very precision that lets our immune system zap specific germs can be fooled by these molecular lookalikes. And thinking about this evolutionarily, there's this idea called the costly autoimmunity hypothesis.
Costly autoimmunity sounds ominous.
It suggests that some parasites might have actually evolved to mimic host proteins as a survival strategy.
Wait, They want to cause autoimmunity? Why?
The thinking is, if the parasite triggers an autoimmune response in the host, that self-attack might actually distract or disrupt the host's immune system, making it harder for the host to effectively clear the parasite. It's like strategic camouflage combined with sabotage.
Wow, that's devious.
It is. But it's a risky game for the parasite, too. The source material notes this trade-off. Causing host damage might help it persist, but a really strong autoimmune response could potentially harm the parasite, too, or cripple the host so much the parasite can't survive. So it's a delicate evolutionary balancing act, maybe favoring pathogens that cause just enough chronic trouble.
Mind-bending stuff. But okay, if we have tolerance, those mechanisms like clonal deletion and tregs, how does mimicry actually bypass them? It feels like the alarm should still be off for self-reactive cells.
That's a great question, and it points to the fact that tolerance isn't absolute. Remember those self-reactive lymphocytes that sneak past central tolerance?
Yeah, APs.
They're out there, usually quiet, maybe suppressed by tregs or lacking the right activation signals. Now imagine a pathogen comes along, and it has an epitope, that small, recognizable fragment that looks very similar to a self-protein, and it triggers a big inflammatory response.
Okay.
The pathogen provides the mimic antigen, signal 1, for T cells. But crucially, because it's an active infection, it also provides strong danger signals, PMPs, which activate those dendritic cells fully. The dendritic cells then provide the essential co-stimulatory signals, signal 2, and release inflammatory cytokines, signal 3.
The 3 signals needed to really activate a T cell.
Exactly. Signals that are normally missing when a self-reactive T cell just encounters self-antigen in a quiet, non-inflammatory state. So the mimic from the pathogen, combined with the inflammation it causes, provides the perfect storm to wake up those dormant self-reactive lymphocytes.
And once they're awake,
They proliferate. They become activated effector cells.
Yeah.
And because their original target specificity was for something that looks like self, they now go out and attack the healthy host tissues that share that similar structure. Mistaken identity, triggered by the infection.
that makes sense. It bypasses tolerance because the context is different. There's real danger and inflammation.
Precisely. The context is everything. A really stark example of how this can work and how broad it can be is HIV-1 mimicry.
HIV, how does that fit in?
Research shows HIV-1 is pretty good at mimicking structures on our own T-cell receptors, the TCRs, and other important host proteins.
It mimics the very cells it infects.
Yes, partly. And this creates a terrible dilemma for the immune system. To fight HIV, it has to generate responses. But if some of those responses cross-react with T cells themselves or other tissues that HIV mimics,
Ends up attacking its own immune army.
exactly, it contributes to the destruction of T cells, which is the hallmark of AIDS. It can lead to autoantibodies that attack lymphocytes and potentially attack other tissues too, altering self-tolerance in a broader way. Some researchers even think this constant immune confusion might contribute to things like gut problems seen in AIDS by changing tolerance to our normal gut microbes.
So the mimicry isn't just causing a separate autoimmune issue, it's actively helping the virus destroy the immune system.
It can be deeply intertwined, yes, a truly vicious cycle driven by molecular deception.
Molecular mimicry targeting myelin, the impact on the nervous system.
Wow, okay, so this mimicry can cause systemic problems, even help viruses like HIV. What does this mean for our bodies in other ways? How does something like this hit the nervous system? That seems particularly scary.
It is, and that's because the nervous system relies so heavily on one critical structure, myelin.
Right, you mentioned myelin earlier, the insulation.
Exactly. Think of nerve fibers' axons as tiny electrical wires carrying vital messages. Myelin is the fatty sheath wraps around them like the plastic coating on a wire.
And that helps the signal travel fast.
Incredibly fast and efficiently. It allows for what's called saltatory conduction, where the nerve impulse jumps between gaps in the myelin. Without intact myelin, those signals slow way down, they get garbled, or they might just stop altogether.
Communication breakdown.
Total breakdown. And that's what happens in demyelinating diseases. The myelin gets damaged, nerve signaling fails, and you see neurological symptoms, numbness, weakness, vision problems, paralysis, cognitive issues. It's devastating.
So how does molecular mimicry lead to attacking myelin specifically? How do scientists study that?
A key tool here is an animal model called experimental autoimmune encephalomyelitis, or EAE.
EAE. Okay, what is it?
It's basically a way to induce a demyelinating disease, usually in mice, that mimics some aspects of human diseases like multiple sclerosis. It gives us a controlled way to see how the immune system can be provoked to attack myelin.
How do they induce it?
Typically, you take immune cells from a mouse and stimulate them in the lab with a myelin protein. Common ones are myelin oligodendrocyte glycoprotein, MOG, or bits of myelin basic protein. You often mix this with a strong immune booster, an adjuvant, to really fire up the response.
So you're training immune cells to recognize myelin.
Essentially, yes, priming them. Then you transfer these activated myelin-specific immune cells into a healthy mouse.
Yeah.
And those cells travel to the central nervous system. The brain and spinal cord find the myelin, recognize it as their target, and launch an inflammatory attack. Within weeks, the mouse develops symptoms like tail weakness, hind limb paralysis, classic signs of demyelination.
What, so you can recreate the attack in the lab?
It's a powerful model, yeah. It lets us dissect the cellular and molecular players involved.
And speaking of players, you mentioned those pro-inflammatory Th17 cells before. Do they show up in EAE? Are they driving the damage?
They are absolutely central players in many forms of EAE. As discussed in some of the research we looked at, like Dan Lippman's work, Th17 cells are consistently found in these situations of tissue destruction and autoimmunity.
And if you prime Th17 cells against myelin specifically,
They are very effective at inducing EAE. Their power comes from that cytokine cocktail they produce, especially IL-17, but also IL-22, IL-6. These cytokines are found ramped up in the EAE model.
what does IL-17 do in the brain or spinal cord?
It's a potent recruiter. It calls in neutrophils and other inflammatory cells into the central nervous system. These recruited cells then contribute directly to damaging the myelin sheath and even the underlying nerve fibers. IL-6 helps drive the Th17 differentiation itself. It creates this inflammatory feedback loop.
A vicious cycle right there in the nervous system.
Exactly. It clearly highlights Th17 cells as major aggressors in this type of autoimmune attack on myelin. And naturally makes them, and the cytokines like IL-17, very interesting targets for potential therapies.
Okay, that's the model.
Yeah.
What about in people? Are there human conditions where we clearly see this kind of post-infection, mimicry-driven attack on myelin?
Absolutely. One of the classic examples, really a prototype, is Guillain-Barré syndrome, or GBS.
GBS, I've heard of that. Isn't that the one where people get paralyzed quite suddenly?
Yes, it's an acute condition. It's an immune attack, a polyradiculoneuropathy that damages the myelin of peripheral nerves, the ones outside the brain and spinal cord. And very often it kicks off after an infection.
What kind of infection?
Often a gut infection, like from the bacterium Campylobacter jejuni, but also sometimes after respiratory infections. The immune system fights off the bug, but then seemingly out of the blue starts attacking peripheral nerve myelin.
And that's mimicry.
It's strongly believed to be. Components of the bacteria, like certain lipooligosaccharides on Campylobacter, look structurally similar to molecules called gangliosides, which are part of the nerve myelin sheath. So antibodies made against the bacteria accidentally cross-react with the nerves.
Mistaken identity again.
Exactly. And interestingly, our sources mention GBS cases linked to SARS-CoV-2 infection and even rare reports following certain vaccines, like the AstraZeneca COVID vaccine. It underscores how different triggers infections, maybe very rarely vaccines, can potentially set off this autoimmune cascade against nerves in susceptible people.
Susceptible people. That seems key. What about other conditions, maybe more chronic ones like MS? Is there a mimicry link there too? No. Or following vaccines?
The picture gets more complex there. The source discussing demyelinating diseases after vaccination does mention reports of GBS, multiple sclerosis, MS, and another condition called transverse myelitis following hepatitis B vaccination, for instance. Now, proving a direct causal link between a vaccine and a chronic, complex disease like MS is extremely difficult. The biological plausibility, though, is considered. The hepatitis B vaccine uses a viral surface protein, HBsAg. It's possible that in a rare, genetically predisposed individual, the immune response to this protein could cross-react with myelin components in the central nervous system, potentially triggering or worsening MS or a similar condition.
So biologically possible, but hard to prove. It's not just coincidence.
Extremely hard. And large population-based studies are really crucial here. Generally, these big studies have not found a significant increase in the overall risk of developing MS after routine vaccinations like hepatitis B. But the concept of molecular mimicry provides a potential mechanism for why these rare individual cases might occur. It's that constant tension between population safety and rare individual reactions.
Understood. Now, you mentioned SARS-CoV-2 triggering GBS. Has the pandemic revealed more about mimicry and autoimmunity in general?
It really has. SARS-CoV-2 has unfortunately become a major case study in virus-induced autoimmunity. Multiple studies have found significant molecular mimicry shared epitopes between human proteins and proteins from the SARS-CoV-2 virus.
So the virus looks like us in multiple ways.
In potentially significant ways, yes. This can lead to widespread cross-reactive B cell and T cell responses. The immune system trying to fight the virus ends up generating antibodies and T cells that can also target our own tissues.
Is this linked to severe COVID?
It seems to be. We see this autoimmune-like antibody response, particularly in severe cases. It might contribute to the dangerous cytokine storms, that massive uncontrolled release of inflammatory signals like IL-6, and also to damage in various organs, not just the lungs, but heart, kidneys, and potentially the nervous system, too.
It's not just attacking the virus, it's causing collateral damage through self-attack.
That seems to be part of the picture in severe disease. And another really striking finding, highlighted in one of the papers, was the presence of autoantibodies against type I interferons in some patients with life-threatening COVID-19.
Interferons. Aren't they our first line of defense against viruses?
Exactly. They are crucial innate antiviral proteins. So developing autoantibodies that neutralize your own interferons is like taking out your own early warning system and initial defense force. It cripples the innate response, likely leading to higher viral loads and much more severe disease.
Wow, so the adaptive immune system making antibodies is actually sabotaging the innate immune system.
That's what it looks like in those cases. It's another facet of how autoimmunity, potentially triggered or exacerbated by the viral infection and its mimicry, can worsen the outcome. It really shows the diverse and sometimes devastating ways molecular mimicry can play out during an infection.
Investigating and intervening the future landscape.
Okay, this is all incredibly compelling, especially the links to real human diseases like GBS and the insights from COVID. But, how do scientists nail this down? How do they definitively prove that molecular mimicry after, say, an infection actually caused the subsequent autoimmune disease? It feels like connecting those dots must be incredibly challenging.
You are absolutely right. Establishing definitive causality is a huge hurdle. It requires going way beyond just finding an association or biological plausibility. There's a set of pretty stringent criteria scientists look for.
Like what?
Well, first, you need a specific clinical syndrome that consistently appears after the trigger, and for which mimicry provides a strong, logical, biological explanation. GBS after Campylobacter is a good example. The timing, the specific target, peripheral myelin, the known mimicry between bacterial components and gangliosides.
Okay, a clear pattern and plausible mechanism. What else?
Laboratory confirmation is vital. Can you actually detect the pathogen, or maybe find those specific cross-reactive antibodies or T cells in the patient that recognize both the bug and the self-tissue? That's strong evidence, though technically challenging.
Makes sense. Anything else?
Ideally, though it's often not ethical or practical, recurrence upon re-exposure, a positive re-challenge, would be very strong proof. If someone got GBS after an infection and then got the same infection again and developed GBS again, that's telling. But you wouldn't deliberately reinfect someone.
No, definitely not.
So perhaps the most crucial piece, especially when considering population-level questions like vaccine safety, is robust, controlled epidemiological studies. Large-scale studies.
Comparing groups of people.
Exactly. Comparing the rate of the disease, like MS or GBS, in large groups who were exposed to the trigger, infection or vaccine, versus those who weren't, controlling for other factors. Do you see a statistically significant increase in the exposed group overall? That's how you assess population risk, moving beyond individual cases or just plausibility. These studies are complex and expensive, especially for rare events, but they're essential for definitive answers.
Right, separating the signal from the noise on a population level, that clarifies the challenge. But okay, even with those challenges, understanding these mechanisms, the mimicry, the TX17 cells, the cytokines that must help with treatment, right? How does this knowledge translate into therapies?
Absolutely. It's not just academic. This understanding is directly shaping treatment strategies, knowing that specific cytokines, like IL-6, fuel the fire in cytokine storms or chronic inflammation points directly to using IL-6 inhibitors. We've seen that used in rheumatoid arthritis for years and more recently explored for severe COVID-19 based on the idea of dampening that hyperinflammation.
Targeting the specific inflammatory signal.
Precisely. And similarly, because TH17 cells and their main cytokine, IL-17, are so implicated in autoimmune tissue damage, especially in models like EAE and conditions like psoriasis, drugs that block the IL-17 pathway are a major focus. Several are already approved and successful for certain conditions, and they hold promise for others where TH17 seems to be a key driver.
So targeted biological drugs are a big part of it. What else?
Well, there are broader approaches, too. General immunosuppressive therapies, like corticosteroids, are still widely used to just calm the whole immune system down during flares. In acute situations like severe GBS, plasma exchange can be used to physically remove the damaging autoantibodies from the blood.
Filtering the blood, basically.
Essentially, yes. But the field is really pushing towards more precision. The idea of targeted elimination of self-reactive lymphocytes is a major goal, getting rid of only the bad actors, the specific B or T cells causing the autoimmunity, without nuking the entire immune system and leaving the patient vulnerable.
That sounds like the holy grail. How close are we?
It's challenging, but there's exciting progress. And this leads to something really cutting edge, adapting CAR T-cell therapy for autoimmunity.
CAR T, like the cancer therapy, how does that work for autoimmunity?
It's the same principle, just repurposed. Remember, CAR T-cells are T-cells engineered in the lab to recognize a specific target antigen. In cancer, they target tumor cells. For autoimmunity, the idea is to engineer them to target the immune cells causing the problem, particularly autoantibody-producing B cells.
So the CAR T cells hunt down and kill the rogue B cells.
Exactly. For example, some very promising early studies have used CAR T cells engineered to target CD19, a protein found on most B cells. In conditions like lupus, wiping out the B cell population, including the self-reactive ones, has led to remarkable remissions in some patients. It's like hitting a reset button on that part of the immune system.
Wow, a reset button.
Yeah, or even more specific approaches are being tried, like targeting only the B cells that make antibodies against a specific self-antigen involved in a particular disease, like the DSG3 antigen in the skin disease pemphigus. The potential is huge, moving from just dampening the immune system to actually eliminating the root cause, the specific self-reactive cells. It's a paradigm shift. Still early days, lots of challenges with costs, side effects, but incredibly promising.
It really sounds like it, a totally new way of thinking about treating these conditions. But this incredible power we're gaining over biology, the ability to engineer cells, understand mimicry, it raises that uncomfortable question, doesn't it? The same science that gives us CAR-RT therapy, could it be misused? Is this the dual-use dilemma people talk about?
It is exactly that. The dual-use potential of biotechnology is a profound and ongoing concern. The same breakthroughs in molecular biology, genetic engineering, synthetic biology that allow us to design life-saving therapies could, theoretically, be twisted to create more dangerous bioweapons.
How real is that threat?
Well, history provides sobering lessons. Think about the scale of the former Soviet Union's biopreparat program. They weaponized agents like anthrax, smallpox, Marburg, even worked on antibiotic resistance and vaccine evasion. That wasn't theory. It was state-sponsored reality, using the best available science at the time.
And today's science is even more powerful.
Much more powerful and more accessible. Technologies like CRISPR gene editing make modifying organisms, including pathogens, faster and easier than ever. Synthetic biology allows building DNA from scratch, potentially even entire viruses. The ability to synthesize pathogens de novo is a game changer.
So you could engineer a virus to be more deadly or to evade vaccines.
That's the concern. Increased virulence, altered host range, resistance to countermeasures, and the worry isn't just rogue states anymore. The dissemination of knowledge and tools means that the capability could potentially fall into the hands of smaller groups, or even individuals with malicious intent. The ease of sharing protocols and ordering synthetic DNA online presents real challenges.
That's deeply unsettling.
It is. Even research done with good intentions, like gain-of-function studies trying to understand pandemic potential, walks this fine line. How do we learn what we need to protect ourselves without inadvertently creating or revealing how to make something more dangerous?
And this ties back to understanding immunity and mimicry, too.
Absolutely. Understanding exactly how pathogens evade immunity or how mimicry works is crucial for defense and treatment. But that same knowledge could potentially inform ways to design pathogens that are better at evading immunity or triggering harmful autoimmune responses. There's even discussion, though perhaps futuristic now, of genetic weapons targeting specific populations. The fundamental science overlaps with precision medicine.
So the power to heal and the power to harm are advancing hand in hand.
They often are, on the same technological edge. It just underscores the critical need for really robust biosecurity, strict ethical oversight of research, international cooperation, and a strong sense of responsibility within the scientific community itself. It's a challenge that isn't going away. outro.
Wow. Okay, what a deep dive that was. We really covered a lot of ground. We started with our immune system as this amazing protector,
Hmm.
and then saw how molecular mimicry can completely flip the script, turning those defenses against us, especially hitting vulnerable spots like myelin in the nervous system.
Yeah, we explore that intricate dance of immune recognition, how pathogens can evolve these cunning disguises, and how the delicate balance of immune tolerance can be broken.
Leading to devastating conditions like GBS, potentially influencing MS, and even playing a role in the severity of something huge like the COVID-19 pandemic.
Exactly. From the basic science of T cells and B cells, antigen presentation, the role of Th17 cells and tregs, right through to the clinical impact, and even the therapeutic frontiers like CRT.
It really makes you think about how deeply connected our own biology is to the pathogens constantly swirling around us. This whole mimicry concept, it forces you to consider that constant interplay.
It truly does.
And it makes you wonder, with pathogens always evolving and our medical tech getting more and more sophisticated, how is this understanding of molecular mimicry going to change things?
That's the big question, isn't it? How will it reshape not just how we treat autoimmune diseases,
Mm.
but maybe the fundamental principles of how we design vaccines, or develop new diagnostic tools, or even how we approach biodefense in the decades ahead?
And tied into that, considering that dual use potential we just discussed,
Yes.
maybe the most critical question isn't just what we learn, but how we make absolutely sure this incredibly powerful knowledge about manipulating immunity, about mimicry, about life itself, how do we ensure it's always used for good and never for harm?
A profound thought, and perhaps the most important one to carry forward.
Definitely something to chew on. Until our next deep dive.